Hello friends, my name is Rizwan Hafeez and today we are going to talk about a very important topic that most of the students get confused about it is the difference between chromosome, chromatin and chromatids. If you stay with me till the end of this lecture, I assure you that you will be having very clear-cut concept related to chromosome, chromatid and chromatin. Clear? In the previous video, I have talked about the structure of DNA. I have talked about homologous chromosome, homozygous organism, heterozygous organisms. We have talked about genes and alleles. If you want to learn more about those concepts, you can visit my lecture. Link is given in the description. Okay. So first of all, let's talk about the DNA before talking about the chromatin and chromosome. We know that the length of chromosome in our cell is basically approximately 1.5 meter. 1.5 meter. Okay, if we spread DNA from one cell, it will be approximately a 1.5 meter. But we know that this 1.5 meter length DNA is present inside the cell. It means this DNA has to be very tightly packed. It has to be very condensed form. Furthermore, it is not only present inside the cell. In fact, it is present inside the nucleus. Inside the nucleus of the cell. Clear? So it means this DNA has to be compressed so that it may reside inside the nucleus of the cell. Clear? Now what happened? This DNA wrap around a special protein that is called as histone protein. Okay, this is basically a packing of DNA. I have also explained about the packing of DNA in my previous video. If you want to learn about the packing of DNA, you can watch my those video. In this video, I will just summarize this concept and I will move toward the chromatin and chromosome. Okay, the link will also be given about the packing of DNA in the nucleus. Okay, so what I was talking about, this DNA is wrapped over the histone protein, and now these histone protein and DNA, it will further condense, they will further come closer to each other, and a structure that is called as nucleosome will be formed. Nucleosome. And this nucleosome, it will further condense, further compact together, and a thread like structure that is not visible with the electron microscope will be formed, and this thread like structure is basically called as chromatin. It is called as chromatin. Clear? Now, before moving forward chromatin to chromosome, we will first discuss the cell cycle. Cell cycle. Okay, and then we will link chromatin with the cell cycle, and then you will be able to having a clear cut idea about the chromosome. So stay with me. We know that, for example, this is our cell. Okay, and here is the nucleus, and in the nucleus we know that DNA is present. DNA is present. Okay, now what will happen if this cell want to divide itself? It want to undergo mitotic division. So first of all, what will happen? Obviously, the replication of DNA. It means after the replication there will be two DNA. Replication of DNA will take place. Then division of nucleus will take place. Then division of division of chromosome, then division of nucleus, and division of cytoplasm will take place. And from one cell, two cells will be formed. Two cells will be formed in this way. So it means the first step is replication of DNA. Replication of DNA. And for the replication, we need specific enzymes, DNA polymerase, heligase, ligase, nuclease, specific enzymes, some protein factors are required for the replication of DNA. So when our cell is producing those enzymes, those proteins which are required for the DNA synthesis, we say that our cell is in G1 phase. Our cell is in G1 phase when it is preparing itself for the DNA replication. When it is preparing itself for the DNA replication, we will say that our cell is in G1 phase. And once all the enzymes, all the protein required for the DNA replication has been formed, now our DNA is going to replicate. It is going to make its copies. And when the DNA replication will start, when the replication process will start, we will say that our cell is in S phase. Our cell is in S phase. Now what will happen? A copy of DNA has been formed. After the S phase, we will be having replicated DNA as well. But still, cell has not been replicated. Cell has not been divided. Only DNA has been divided. Now what we need to do? Now chromosome need to be divided. They need to be separated. Clear? Nuclear material need to be separated, and whole cell need to be converted into two daughter cell. So it means for the separation of chromosome, for the separation of nuclear material, for the separation of cytoplasm, we again need some specific enzymes and some specific proteins. And when the cell is preparing itself for the division of cell, for the division of nuclear material, for the division of chromosome, for the division of cytoplasm, when cell is preparing itself for division of all these things, we will say that cell is in G2 phase. It is preparing itself for the mitosis. Mitosis is basically division. Mitosis is basically division of cell into two cells. But when it is preparing itself for the mitosis, we will say that it is in G2 phase. And when the process of mitosis will start, when the process of division will start, we will say that the cell is in M phase. And we know that mitosis itself has stages that include prophase, metaphase, anaphase, telophase. Okay. So in today's video, we are not going to talk about the mitosis. In the next video, we'll talk about mitosis and its stages. Well, from G1 to G2 phase, G1 S phase and G2 phase, they are collectively called as interphase. Now you will be worrying about that you are explaining about chromatin. Now why you are explaining interphase and all these things? They are basically linked with each other. How? During the interphase, G1 phase, S phase and G2 phase, this chromatin, the condensed form of DNA and histone protein, it will be present as such. It will be present as such. But when the first stage of mitosis will start, this chromatin material, it will further condense itself. It will further condense itself to form this structure. And this structure is basically called as chromosome. So during the interphase, DNA and proteins, they will be called as chromatin, and during the mitosis, or we can say prophase, this DNA material will further condense, further compact to form this chromosome structure, and now chromosome will be visible. Clear? Okay. Now, what is chromatid? What is chromatid? Obviously, we know that the sister chromatids in one chromosome have two chromatids, but I think you will not have a clear-cut idea about the chromatids. Now, let's talk about what are chromatids. To understand the concept of chromatid, let's talk about homologous chromosome. In the previous video, I have talked about, but I will just give you a brief introduction. We know that in our cell, there are basically 23 chromosomes are from the mother side and maternal side, and 23 chromosomes will be from the paternal side. Clear? Now, the chromosome number 1 from the maternal side and chromosome number 1 from the paternal side, they are basically homologous to each other. They are basically homologous chromosomes. Clear? Chromosome number 9 from the maternal side, chromosome number 9 from the paternal side, they will be homologous. Chromosome number 5 from the maternal, chromosome number 5 from the maternal side, they will be homologous in one cell I am talking about. So it means all chromosomes are basically having a homologous pair. Clear? I have also explained that these 23 chromosomes are basically N. We donated with N. Similarly, these 23 chromosomes will also be written as N. And you know that we have 23 number chromosome from mother and 23 number from father. So our cell will be 2N. Listen this point very carefully, if you want to understand the clear concept of chromatid. Similarly, the amount of DNA is shown by capital N. Capital N. And when there are 23 chromosomes from the mother side, it means they will be having N chromosomes, N number of DNA, N amount of DNA, and 23 chromosomes from the paternal side, they will be having N amount of DNA. So it means the amount of DNA is also 2N. Amount of DNA is also 2N. Clear? Now let's talk about one homologous chromosome. Stay with me. We are going to talk about a very important concept. For example, this is chromosome number 1 from the mother side and chromosome number 1 from the father side. So it means they both are homologous chromosomes. They will also be having a kind of DNA. DNA in it. Clear? Now, when the process of replication will start during the S phase, this DNA will divide itself. This DNA will divide itself, and another copy of chromosome will be formed. But this chromosome will be attached on the same centromere. The position at which two chromosomes are attached with each other is called as centromere. Okay? This is basically primary construction. I will talk about this centromere in some other video. So, similarly, after the S phase, this DNA will replicate and another copy of chromosome will be formed. But this chromosome will also attach on the same centromere. Getting me? Clear? So it means after the replication, after the S phase, this double structured chromosome will be formed. They are not two chromosomes. This is just one chromosome. How? You will say, sir, replication of DNA has been taken place. So we should say that they are two chromosomes. No. The number of chromosomes is defined by the number of centromeres. As they are attached on the same centromere, so we cannot say that there are two chromosomes. Centromere is one, so we will say that this is one chromosome, but this one chromosome is double structured. The initial chromosome was also single chromosome, but it was single structured chromosome. This was a single structured chromosome. But there are also one chromosome, but this is double structured chromosome. They are double structured chromosome. Clear? Getting? Okay. Now, in a double structured chromosome, these two are basically called as chromatids. These two individual chromosomes will be called as chromatids, or we can say that both this chromatid and this chromatid, we can say that they are basically sister chromatids. Sister chromatid, because they are from the same chromosome, same centromere. So they are basically sister chromatid. But this chromatid and this chromatids, they are basically non-sister chromatid. Non-sister chromatid. I think now you are very much clear about the chromatin, chromosome, and chromatid. After the DNA replication, the sister chromatid will be formed, and chromosome will be converted into double structured chromosome. And DNA will be, for example, in this cell, when the replication of DNA has been taken place, two chromosome division and DNA division has been taken place. Now let's talk about, for example, there are 23 chromosomes. So n plus n, there are 2n chromosomes. But if we talk about the DNA, there are 1, 2, 3, 4. There are 4n. Okay, 23 and 23. 2n from the mother side, 23. 2n from the father side. So after the S phase, the DNA will be 4n, while the chromosome will be 2n. Okay, I think now you are very much clear about the chromosome, chromatid and chromatid. So it means when our DNA, our chromosome will enter into the mitosis, it means S phase has been passed away. After passing the S phase, it will enter into the mitosis. So it means when chromosome will enter into the mitosis, M phase, the chromosome will be double structured chromosome, because after that phase, the chromosome will be converted into double structured chromosome. So this was all about the chromosome, chromatid and chromatin. I hope you must like my lecture. Please don't forget to like and share this video, and don't forget to subscribe my channel so that you may get notification for the future videos. Thank you so much.